Welcome to a new chapter of Glioblastoma, aka GBM, presented by the Glioblastoma Research Organization. This season we're adopting a documentary-style format to offer you a never-before-seen look into the world of glioblastoma from around the globe. Join us as we venture into labs and companies worldwide, providing a comprehensive view of some of the cutting-edge efforts to combat GBM. Through intimate interviews and detailed on-site explorations, we'll offer an insider's perspective on the innovative strategies and real-time progress in the fight against glioblastoma. Thank you to our sponsors whose generous support has made this season possible. Please note that the sponsorship of this season does not constitute an endorsement of any sponsor's products or services. Remember, the information presented is for educational purposes only and it is not intended to treat, diagnose, or prevent any disease. Always consult your medical provider and healthcare team with any questions.
So my name is Frederik De Smet. I'm a professor at the University of Leuven in Belgium, and I'm the coordinator of the GLIOMATCH Project, which is an EU-funded project to tackle various challenges in the treatment of glioblastoma. So GLIOMATCH is an EU-funded project that was started about a year and a half ago now. The key goals are to improve the use of immunotherapy for brain tumor patients. So far, glioblastoma has had a devastating outcome, so there is almost no cures available. But this is also due to the lack of understanding of the underlying biology of this terrible disease. And so GLIOMATCH aims at a better understanding on how these tumors are composed, so actually in the brain of a patient, but not only at the tumor level, but also understanding how these tumor cells interact with cells from the immune system, which are present in what we call the immune microenvironment of a tumor. And so GLIOMATCH is on the one hand trying to understand how this immune environment is important to apply immunotherapies for glioblastoma, but also we are trying to better understand how patients are responding to immunotherapy. Immunotherapy comes in many different flavors, and when you apply it to a patient, you're not always sure what the outcome is going to be of this treatment. And so GLIOMATCH tries to form an answer to those really big challenges in our domain. Now, another key target of GLIOMATCH is to integrate these insights also with a method that would allow physicians to understand what's going on in a patient while they are on treatment. Of course, we're working with a tumor that is in the brain, and it's not simple to collect material or tissue, some kind of biopsy material from the brain. Nowadays, follow-up of patients is done through non-invasive imaging, for instance, through MRI scans. But of course, the amount of information that can be collected from an MRI is very limited. And so GLIOMATCH wants to integrate the insights that we obtain at the level of the tissue, so really inside the tissue, which cells are there of the tumor, which cells are there of the immune system, and link this to MRI images so that we can better understand from a future MRI scan what is actually happening in the tissue. And this will, of course, allow our clinicians to better understand whether a treatment is actually working and whether a treatment should be adapted to an individual patient. So if you see that a treatment is not working, that they can still change some of it. But this can be done then in a non-invasive way, meaning that a patient can undergo an MRI scan every so many weeks, so that you can really follow up how a treatment is affecting or working in a patient. The biggest inspiration for GLIOMATCH originated from the interaction I have on a daily basis with my colleagues in the hospital, with my colleagues in other research laboratories, where we all try to understand brain tumor biology and how we could improve the treatment for brain tumors. Now, immunotherapy has been a topic that has been very hot and has been really revolutionary for some cancers. Unfortunately, for brain tumors, this has not really worked. So we thought that bringing together experts across Europe, both from a clinical side and from a research side, to really tackle some of the key challenges of glioblastoma, would actually be, could be a game changer for our field. And so, as such, we defined different targets to study in our study. So the patient experience for a patient that unfortunately got diagnosed with a brain tumor starts off with arriving in the hospital, typically with very non-specific symptoms such as nausea, headaches, disequilibrium, things like that. And then they will undergo a variety of tests, being non-invasive imaging, so an MRI scan will be taken, there might be some blood draws being done. Unfortunately, then often when a brain tumor is diagnosed, the tumor is often in a very advanced stage. Certainly for a glioblastoma, the tumor typically does not give a lot of symptoms in the early phase and is only discovered when the tumor is already in a late phase. So once that's discovered, then a surgery will be scheduled to remove as much of the tumor tissue as possible, following which the patient will get a little bit of time to recover from the surgery, and then they will start with a very intense program combining chemotherapy together with radiation therapy. This will last for a few months, and then after finishing these cycles, there will be a wait-and-see policy where the patient will have to come back to the hospital about every 3 months for some non-invasive imaging. Unfortunately, for the majority of patients, the tumor will return within 1 year after diagnosis. And then, dependent on the state of the patient, this whole process can start over again. So there might be another surgery, there might be another post-surgical treatment. And then we are talking of a track that lasts, let's say, between 2 and 3 years maximum. What we define as key success hallmarks of a project like this is that, first of all, you need to have a very clear collaborative environment. So in GLIOMATCH, we work together with 15 different partners who all bring in their own expertise. But of course, to make this project a success, we have to make sure that all these partners will actually work together. So this is where my role as coordinator comes into play. And of course, it's very important to make sure that everybody is aware of the key aims of every part of the project, how they have to, which partners of the project have to work together to make the magic happen. And so my role as coordinator is to ensure that everybody meets the predefined aims and challenges of our project. To keep the voice of the patient central in this still very fundamental and to some level translational project is by, first of all, one of the partners of GLIOMATCH is a patient organization called Cancer Patients Europe, CPE abbreviated. And they are involved in multiple of this type of consortia. They also have a voice within, for instance, the European Parliament, where a few months ago they have been able to already voice some of the concerns that they have to ensure that regulation, policy, and that the voice of the patient is actually resonates within the Parliament. Now, of course, within this particular project, we make sure that the patient is by default central in what we do. The goal is, of course, to improve the outcome of the patient. We are starting up a variety of prospective trials, meaning that these will be trials where new patients will get the opportunity to be treated with some new, very novel, really state-of-the-art therapies. Of course, there are no guarantees that these therapies will already work as they are now, but it's very important that patients can really actively participate in different parts of the GLIOMATCH project. So we're now about 1.5 years down the line in the GLIOMATCH project. A big part of the project has now been aimed at installing all the required legal and ethical approvals, that we are allowed to work with patient materials, that we are allowed to work with patient data. That's of course very important to have a proper legal framework, because also with all the data that we will generate, we want to make sure that we can also use this as leverage to align with other existing initiatives so that GLIOMATCH in its own can actually work as some kind of multiplier for other projects. For instance, we're also already talking to some companies who are developing new drugs to see if there are any data that we are generating could already improve how their drug can be further developed. Now, so all the ethics and legal documents are nearly in place. In addition, we have been working very hard on further fine-tuning all the state-of-the-art technologies that we are applying to really understand what are all these different cells inside a tissue of a glioblastoma tumor. And this, of course, also requires still some adaptation and implementation of really state-of-the-art technologies, because many of these technologies that we are applying, they're very recent. They keep on evolving, so we really try to implement always the newest of the newest and make sure that the insights that we will be able to generate for all these patients is driven to the maximum. So the primary goals at the moment for the GLIOMATCH project is, first of all, to make sure that we can finalize our in-depth analysis of all the patients initially of retrospective studies. So this means patients that have been treated with immunotherapies in the past, that we really understand what is going on, what was the effect of the treatment in each of these patients. This will still keep us busy for at least another year and a half. But in parallel, we're also starting up prospective trials, so where new patients who are currently suffering from their disease can also be enrolled and really will be able to get access to some completely novel treatment schemes so that we can investigate also what the effect is of these treatments on these individual patients, but also if we see that some of these treatments are working, they will get prolonged access to these treatments as well.
I am Steven de Vleeschouwer. I am trained as a neurosurgeon and currently working for 20 years as a staff neurosurgeon in university hospitals in Leuven, where I have an affiliation at the university in the Department of Neuroscience in a professorship. And my main clinical but also research interest is in oncological neurosurgery, meaning that actually we perform operations where brain tumors of any kind are being removed in the most safe and efficient way as possible. And in terms of lab activities, I have been working for more than 25 years now in a research line where we try to understand the importance of the immune microenvironment of the brain tumors, which is a very particular area of interest because it's a condition we should understand in a better way before we will be able to implement efficient immunotherapies for brain cancers, which still isn't the case today. Well, the care of as such has changed in terms of the technology that has become available to actually perform safe and efficient surgery. So the aim of surgery, of course, is to remove as much as possible of the brain tumor without jeopardizing the functions of the patients. So you have this double objective. The oncological objective is actually to remove as much tumor as possible, and the functional objective is to keep your patient functionally intact, to make sure that the quality of life of the patient is not hampered and damaged after the surgery. So these two purposes actually are obtained because of the fact that we do have new technologies helping us in achieving these goals, both of them basically. In terms of removing the brain tumor, we now rely more and more on fluorescent-guided surgery, which means that actually we give a kind of prodrug to the patient before he or she is being operated. The prodrug is being metabolized in the brain, or especially in the brain tumor. And during the surgery, when we use the proper light and the proper settings of our operation microscope, we are able to recognize the fluorescent areas in the brain which actually coincide with the pathological parts that have to be removed. On the other hand, we want to monitor the functions of the patient, like motor functions, sensitivity function, visual function, linguistic functions. All these functions can be monitored during surgery, either in a purely electrophysiological way or in terms of, for instance, language monitoring. We wake up patients while operating on the brain tumor to make sure that we do not cause permanent damage of the particular function we want to preserve. And the combination of these new technologies made the brain surgery for brain tumors safer, but more efficient as well. I can explain what actually surgery adds to the research perspective of GLIOMATCH. First of all, it's a starting point, because of course we need a tissue diagnosis before being able to select the proper treatment of the patient. But in terms of research, we actually also use the tissue not just to confirm the diagnosis, but to understand the outstanding questions in terms of how is the microenvironment, how are the cells behaving, what's the infiltration pattern of these cells, which molecular particularities do these cells have, and all of this actually requires tissue to be interrogated in typical research assays in the lab. So actually we are the first step, providing researchers with proper samples of the brain tumor in different locations, around blood vessels, in infiltration areas, to actually fully understand what's going on inside the brain. So it is, first of all, delivering proper samples for examination and interrogation. And on the other hand, of course, we are following up on these patients, and we are building clinical databases which we want to correlate with the tissue examinations of each single patient. And in that way, we can start building correlations between what we see in the clinic and how the tumor and the patient behaves in a clinical course of diagnosis and treatment on the one hand, and how the tests in the lab explain this course of disease. So we are actually, I think, a link between the research and the clinical course of the patient. So the question is, what are the biggest misconceptions of patients regarding brain surgery? And I think there are a few. The biggest misconception probably is that people tend to believe that any type of brain surgery will result in some kind of loss of function. And this is, of course, a logical concern, because of the fact that our brains, they actually orchestrate our whole being and thinking. But it is perfectly possible, if you understand the anatomy and the physiology of the brain, to actually work and operate in the brain without leaving permanent damage. The only thing is to make sure that you take all proper precautions not to damage the crucial parts. And this is a misunderstanding. Many patients actually, they start from the thinking that if they go into brain surgery, they will come out of surgery with some deficit whatsoever. And I think that's a misconception. We can do a lot of things in the brain without causing permanent harm. Of course, you can't do anything, but you can do a lot of things. The role of tissue sampling in research, especially for brain tumor research, I think is many. First of all, I often hear the remark from patients that they are happy, that they are able to contribute to the research perspectives, because many of them realize that they are suffering an existential disease for which not too many alternative treatments are available. So many people realize that research is the only way to make progress, and most of the patients are happy to be able to contribute to that. So it's a kind of connection with the patient and the research, and many of them love this. Second, the tissue is key in anything we learn from this, because You probably aware of the fact that we are able to scan patients in different types of scans and learn a lot of indirect things from what we see on images. However, the ground truth of what's going on in the brain and what the pathology of a brain tumor typically means for these patients can only be fully understood if we dive into the tissue level, where cells interact with the brain and with the normal brain cells like neurons and glial cells and blood vessels. And they all have an interaction which we cannot fully understand just by indirect interrogation of the images. So the tissue is key to really understand the basics of where we should tackle this disease. That's exactly connected to both Abhishek's and Paul's work. Paul is, of course, an oncologist, a medical oncologist, so actually we are treating the same patients. Once we have operated on the patients, we transfer them to the oncology department for the post-operative treatment, which is taken care of by Paul and colleagues on the one hand. And Abhishek, of course, is running the immunology research lab, where he really dives into immunological signatures, cell signatures of those immune cells that you can find in the brain, the normal brain, but especially in the brain tumors, and which try to interact with these pathological cells, initially to control them, but very often, they are being recruited as kind of collaborators for the brain tumor. And this is something we start to understand thanks to the work of Abhishek and colleagues in the lab, interrogating the immune system and the interactions with these tumor cells. Yeah. Well, the relation of state-of-the-art treatments and the more experimental treatments, like immunotherapy for brain tumors, it's indeed, as you mentioned, a balance in the way that we are just starting to understand which patients might qualify best for these types of treatment. Because what we learned in the probably 2 to 3 decades ago, when we applied several types of immunotherapy to brain tumor patients, is that some of them actually react very well to these types of treatments. And for those people, it might be the start of a very long, sustainable disease control. And for others, actually, the immune therapy doesn't seem to have the power to change the course of the disease. And the explanation for this actually is to be searched and found in the characteristics of a patient's immune system on itself. We think the immune system, like many systems, is completely alike in every patient. But what we learned is that there are so many aspects of the immune system that can go either way. And one way will be beneficial for the patient, the other could be detrimental. And actually start with a proper stratification of patients based on their baseline immunological status. This is something that we take in the balance of a decision whether or not a patient might qualify for certain types of immunotherapy. Having said that, in the near future, we believe that immunotherapies for brain tumors probably will be beneficial, especially for patients in whom we can reach some kind of what we call minimal residual disease status. And this means patients in whom we can take the full macroscopic tumor out of the brain, or at least a very large part, in order to give the immune system the chance to recover before we start hitting it with immunotherapy approaches. So let's say personalized medicine treatments that are being developed at the moment, because that's the question, actually can be probably divided in a few different approaches. One of them is immunotherapy. And why is this personalized? Because actually we want to use the patient's own immune system and direct it towards its own tumor cells. So this is a fully what we call autologous system, where we influence patients' immune cells. Often we do harvest these cells upfront, we modify these cells, and we re-administer them. So these are personalized treatments because of the fact that the patient is actually the donor of his own immune cells. That's a very personalized approach, where off-the-shelf applications, as for now, are not really the most promising solutions in terms of immunotherapy. Another way in which we try to steer these new approaches is by what is being called targeted therapies. And targeted therapies, unlike chemotherapy, are actually drugs or compounds that act on a very specific molecular abnormality of the brain tumor, which is not to be found in normal cells. So these targeted therapies, they have the advantage that they probably have less side effect or less collateral damage in the normal tissues. But the downside of it, of course, is that the molecular landscape of these tumors is very diverse if you consider them over a full group of patients. So there probably are no two patients with exactly the same profile. And that's why it's a labor-intensive approach, which is very tailored, but as for now, not really easy to scale up. So discussing and mentioning hope and honesty and balancing hope and honesty when we discuss the diagnosis for patient is of utmost importance, because both components, I think, are absolutely crucial. First of all, the disease is such a threatening experience for the patient that they, of course, have the full right to be informed about the existential problem that a brain tumor very often causes for them. In that way, they, of course, immediately realize that they are hit by a serious disease. And this honesty is very important to gain trust of patients. And, of course, you need patients' trust and confidence in order to make sure that you have an open perspective to discuss all the options. And especially in discussing all the options, there is the hope you can bring in. The diagnosis, of course, is the honesty. But the hope you can bring in is the fact that although this is a very serious disease with a very bad prognosis on average, it has to be said that a few patients, and we do not fully understand why, but a few of them actually fare quite well with very long periods of disease controls under state-of-the-art treatment, but also under some form of experimental treatments. And the hope actually exists in the fact that we do not know upfront who will be actually a successfully treated patient and who will fail in his treatment. So it's not a guarantee at all, and at any time there is some kind of uncertainty remaining as a possible threat for the patient. But there clearly is hope, and we should not abandon hope from patients, because of course from a mental point of view, if you need the energy and if you need the strength to go and fight this disease, you at least need to have some realistic hope to be in the good group and at least try everything that is possible. And that intersection of honesty and hope is crucial for I would say, a patient-doctor relationship anyhow.
So my name is Paul Clement. I'm a medical oncologist with a special interest in neuro-oncology. So I'm treating people with brain tumors. That means all types of brain tumors, but the most common is glioblastoma. And this is also the subject of the GLIOMATCH project, where we want to put some steps forward in research, but also ultimately, of course, in helping patients with this very difficult to treat disease. So when we see a patient with newly diagnosed glioblastoma, the patient is typically referred to the radiation oncologist after surgery, and to me, the medical oncologist, for chemotherapy. This consists of temozolomide. It's a pill, a type of chemotherapy that we combine with the radiation therapy during about 6 weeks, where the patient is taking this pill every day, also during the weekends when there is no radiotherapy scheduled. And then we continue with that treatment 5 days in a month for about 6 months. So that is the typical treatment that a patient would receive with glioblastoma. And with this treatment, we do see only a minority of patients that benefits from it for multiple years, but unfortunately, the majority will not survive more than 2 years. So currently, we are still looking for major improvements in the treatment of this disease. During the last 2 decades, there has been not a lot of progress, unfortunately, in the treatment of glioblastoma. Although there have been many drugs that have been tested, among which targeted treatments and also immunotherapy. But unlike other cancers, in glioblastoma, these were not successful, and there are many reasons you could think of. One of the reasons is that the drugs need to enter the brain, so the brain is protected from toxic agents, and so many of these drugs are not entering sufficiently into the tumor tissue. And with regards to immune therapy, there may also be an issue that immune cells are not always present in the immediate vicinity of the tumor. Of course, and as you know, there exist diseases, immune diseases in the brain, like multiple sclerosis. So it is certainly possible that immune cells enter the brain. So there might be a subgroup of patients that really can benefit from immune therapy, but it is very hard to identify this group of patients. And if we just look at the entire population, the mainstream immune therapy that is quite successful in, for example, melanoma or in lung cancer is not successful in glioblastoma. And that is because only few patients probably benefit from that treatment. And still, we have observed quite impressive results in a few cases. And it's always difficult to tell whether that is just by chance or whether there is indeed a mechanism that may explain why these patients are benefiting from this treatment. And that is the subject of the GLIOMATCH protocol. So to learn more about successes that we have seen in the past, not only with us, but in many big centers. And so by learning from these cases, we would like to see certain characteristics in these patients that might predict what patient would benefit from, for example, immune therapy. Decision-making is a difficulty in patients with brain cancer. One of the reasons why that is that in brain cancer, of course, you cannot easily do repetitive biopsies. In the brain, this would be quite risky to do repetitive biopsies because you have to penetrate the skull. So that is something that is not done, of course, and that is why we rely on imaging techniques like MR scans. And this is true for adults, but also for children, and maybe even more so because the tumors in children are often located in even more difficult spaces, parts of the brain that are very difficult to get to in a safe way, even for diagnostic biopsies. The problem with MR scanning is that it is only, I would say, a shadow of reality. So we need to interpret this data that we see on MR scans with caution. And this is something that is quite known, actually, that particularly in the early weeks after treatment, that images could reflect what we call pseudo-progression, so not true progression, but an image that mimics real progression. And this is a problem because we could make wrong decisions. Maybe we judge that the patient is not benefiting from treatment, whereas it is actually a false image. And so this is something that we learned, and still we do not have perfect techniques to define that 100% sure. So it is based on a lot of factors, some of which are experience, of course, but also some biomarkers. Biomarkers that make it more likely that a pseudoprogression could happen, for example. And of course, there are also clinical parameters that we use to judge this. And of course, we have a knowledge about the time window in which these pseudoprogressions generally occur. And it is thought that, for example, with immune therapy, it is more often the case than with other treatments. So this is a major issue when we are looking at response to treatment and whether it would be in the best interest of the patient to continue a certain treatment. So pseudoprogression is a phenomenon that relates to the evolution on MRI imaging. So when we look at MRI, the patient will receive a contrast-enhancing agent, and this could reflect tumor growth when we do see more contrast enhancement in the tumor site. However, after treatment, this could also be caused by inflammation, which we often see or would like to see even after immunotherapy, but also we can observe this kind of changes after chemoradiation. In some cases, this is more often happening. For example, in certain situations where you have hypermethylation in the tumor, but we are certainly looking for other details, characteristics at a molecular level that could help us tell whether a certain image indeed reflects true progression or not, because you can imagine that is very important in the decisions that we have to make. So in the GLIOMATCH protocol, we are aiming to really make a hub for MRI imaging that is based on a big number of patients that have been treated in the past, and some of which have an exceptionally long response, that have been treated with all kinds of different experimental treatments, among which also immune therapies. And by analyzing these images and specific characteristics and relating that to the biology of these tumors that we also study, we hope to improve our capability to make sure that a certain image correlates with either true progression or pseudo-progression. And so if we are better in characterizing these images, then we also could provide this technology to other centers to help make the right judgment about the evolution of a disease based on the imaging. The ultimate goal of GLIOMATCH, of course, is that it will impact the lives of patients. So it would not merely give only more insight in the scientific background and the biology of the disease, but ultimately we should be able to personalize treatments. Particularly the focus would be on immune treatments for this disease, and that we could identify certain characteristics in patients that would predict that a certain immune therapy would be efficient. And if we would succeed in that, then we might be able to have a real impact on the disease in at least a certain percentage of these patients, and otherwise. And also, we would not start a treatment that would not work in another part of patients, and also with that avoid potential side effects. So the better we can predict whether a treatment would be beneficial, the better we would be able to treat and help patients.
My name is Sandra Jacobs. I'm a pediatric oncologist here in the University Hospital of Leuven. Well, pediatric high-grade glioma, we know that most of them, more than 70% of pediatric high-grade glioma, consist of diffuse midline gliomas, which have specific genetic alterations in their tumor cell. And so in that way, they are completely different from their adult counterparts, which don't harbor these specific genetic alterations in the tumor cells. Most of those diffuse midline gliomas, they are located in the brainstem, which makes them not suitable for surgical resection. So that means these tumors are located in a very difficult part of the brain, where we know that touching the brain can cause severe damage to the patients. So resection is not possible for those patients. Nowadays, we try to have biopsies of the tumors, because the information that we get from the biopsies can help us in further guide the treatment for the patients. The difficulty there still is that those biopsies are very, very tiny, but we try with a very few material that we have to gather as much as information as possible to further steer the treatment for the patients. But in general, it's only radiotherapy that can prolong the life of our patients. And radiotherapy is currently the only proven life-prolonging treatment for DMG patients. We additionally try to improve the treatment and survival for those patients by increasing and more tailor the treatment based on the molecular alterations that we find in the tumors. In addition, we know that those tumors, and there is an increasing interest for IO treatments in those patients, and we hope by improving these treatments that we can better and that we can find better treatments for our patients. So we know that the clinical course for these patients is mostly very short. They suffer from, when the tumor is, for example, located in the brainstem, from cranial nerve palsy, or they can develop ataxia. Sometimes when the tumor is located in the other parts of the brain, patients can present with paralysis or with epilepsy. And then when possible, we try to remove the tumor, but as said, this is not possible for patients with a pediatric high-grade glioma, which is located at the brainstem. And then we try to have those tiny biopsies to gather more information on the genetic alterations in the tumor. And so we still need to face that pediatric high-grade glioma is still, for the majority of the patients, a lethal disease, and we do not have any curative treatments so far. But we try to prolong the life for our patients as long as possible. But unfortunately, the only proven life-prolonging treatment for pediatric high-grade glioma patients is radiotherapy. So most of the patients, they will all start with radiotherapy, and then we will add some kind of targeted therapy or chemotherapy based on the molecular information that we get from the biopsy of the patients. So as the majority of high-grade gliomas in children, they are DMGs, and they have really specific genetic alterations, which we don't find in the adult counterparts. So they are, on the biology point of view, they are completely different entities. So it's really important that we also focus our research on the pediatric high-grade gliomas, and so that we develop models also to study those pediatric high-grade gliomas. Because of this different biology, it's really important not to rely on the information that we get from adult glioblastoma. Well, since pediatric high-grade glioma is still a lethal disease, we do not have curative treatments for our patients. It's really important that we improve the research to better understand the disease and to also better understand the disease, but from that also try to guide and steer patients more in specific treatment regimens, and so better select which patients might benefit from which therapies. And so with GLIOMATCH, we hope with this consortium that we can have a large cohort of pediatric high-grade gliomas where we can better study this disease and from that learn and tailor further the treatment for our patients. Well, since we don't have any curative treatment options for our patients, it's incredibly difficult to face parents with the fate of their children. And so it's important that we really have research done to better understand the disease and improve our treatment for our patients in order to hopefully in the future find any curative treatment for those patients. Since this is still a very, very rare disease, I think it's really important to unite research, to unite knowledge, and to work with big consortia to gather more information, to exchange information between different research teams. And so with GLIOMATCH, we are a very nice consortium, and from that, we hope to better and to better understand the disease and improve the outcomes. But I think we can only achieve this by uniting forces all together.
So I'm Thierry Voet. I'm a bioengineer from training. I'm also full professor here in the University of Leuven and director of the KU Leuven Institute for Single-Cell Omics. And we provide technology support and co-development of technology in the context of glioblastoma research together with Professor Frederik De Smet in this project. So to explain what single-cell omics is and how it can help us understand glioblastoma, I first set a step back and explain that there are, in normal human body, there are nearly 100 trillion cells, individual cells, which belong to a different set of cell types, each also with different functional states. And these cells need to work together in order to exert organ function, tissue function. And similarly, as in a human body, also tissues, disease tissues like cancers, they are composed of a complex ecosystem of normal and aberrant cells. And we need to understand how these communities work together. And for that, we use single-cell omics, where we disentangle that complex ecosystem to its functional units, biological units, being the individual cells. And so with single-cell omics, we read out and characterize the molecular profiles of these individual cells to tell us who they are, what they are doing, and how they are working together. So analyzing individual cells over bulk analysis has a tremendous amount of advantages. With bulk analysis, what we typically do is take a bunch of cells, hundreds to thousands of cells that you put into a single tube. And then you mix all of these cells together. So you lyse them, you mix all of their contents together, leading to a mixture of molecules, which you can then read out. And then, for instance, if we are looking at gene expression, what we typically read out is the average gene expression that is present across these many cells that are present in that bulk sample. However, with single-cell analysis, we can really home in and provide a lot of granularity in what every cell is doing exactly, and so what the exact molecular profile of every cell is in that tissue. So to further understand resistant mechanisms, single-cell omics has really a number of advantages. So with single-cell omics, we can read out when we provide a drug treatment to a community of cells, how these cells are reacting to the drug. And these could be differences in cellular abundance. Some may disappear, some may stay equal, and others may proliferate. And we can also read out their molecular changes. So some cells may start developing altered behaviors where they become drug tolerant to the drug. And so with single-cell omics, we can really read out the mechanisms why cells are changing and how they are changing. And so learn us more about basically molecular and cellular mechanisms that result in drug tolerance or drug resistance. And by doing so, we can also home into novel druggable targets, as well as potentially better patient stratification schemes if we are better understanding how cells react to the drug. So within the context of the glioblastoma project, we are developing new technologies where we are able to read out different omics layers of individual cells or communities of cells in a spatial assay. So when we look at cell identity, this is really determined on the one hand intrinsically between a different, basically a complex interplay between a molecular hierarchy ranging from DNA, leading to gene expression, to protein formation, and also metabolites. And on the other hand, cell identity is also determined by cell-cell communication. And so within the context of the GLIOMATCH project, we are developing new technologies where we can read out basically gene expression as well as chromatin information in a spatial context, allowing us to really understand how cellular communities are built up and their molecular identity in multi-omics layers, and leading to a better understanding of cell identity. The biggest changes and the major leaps forward in single-cell analysis are expected to be to integrate as many layers as possible, as I explained, for DNA, RNA, protein measurements, metabolite measurements, and so on. Currently, we are still limited to a few of those molecular layers that we can identify on single-cell resolution. So there will be an expectation that more of these modalities can be read out from a single cell, as well as movement towards live-seq applications, where you basically, in a continuum, can measure over and over the same cell, leading to better understanding also of drug resistance mechanisms, drug tolerance mechanisms, as well as to multimodal analyses of spatial compositions of cells. Every day there is a new discovery that we make. There are very surprising things that we are continuously identifying with these new tools at hand. Basically, with using the latest stages and the state-of-the-art of these techniques, what this is enabling is that every tissue that we analyze basically has a story to tell. And basically, the analysis that we do by disentangling the tissues to individual cell resolution, and also by putting back together in a spatial context, each time we get a different story, and it's an exciting story that we obtain. So GLIOMATCH is really a catalyst, I would say, in driving research in glioblastoma further. On the one hand, of course, the assays that we develop cost a lot of money, and so the funding on its own is already a plus. But on the other hand, it also brings together a consortium of multidisciplinary experts around internationally that enable us to work in synergy, where we are all pushed by the research questions in each of our disciplines, leading to a synergy that enables to create magic where 1 + 1 becomes 3.
I'm Professor Abhishek D. I'm a tumor immunologist at the Cell Stress and Immunity Lab here at KU Leuven. We study cancer immunology from the angle of both experiments as well as patients. And my role in GLIOMATCH is to take all the data coming from the partners here and try to do functional interpretation of the data from immunology angle so that we can create insights meaningful for the patients. The main goal of the immunotherapy is actually to use our own immune cells to attack the cancer cells and eliminate them. And this works very well for many different cancer types, but has not quite worked very well in glioblastoma patients. This is because for other cancer types, you already have a certain momentum in the immune system available in these patients, which is then exploited by the existing immunotherapies. This momentum does not exist in glioblastoma patient already. This is where the DC vaccines come in. They use the patient's own information and integrating it into this vaccination format, and then allows to create this momentum in these patients where none existed before. So what is more difficult to work with glioblastoma than other cancers is because glioblastoma, as I said, does not have a previous immune momentum. What does that mean is it does not have those distinguishing factors that makes glioblastoma tumorigenic enough for our immune system. So it escapes the eyes of the immune system because it hides this information. This is why it's so difficult to treat glioblastoma, because in other cancers, this information is more easily accessible to our immune cells, but that is not the case in glioblastoma. So we are currently busy trying to use very complex data to understand very basic principle, which is in this case, how is an immune cell sensing the glioblastoma cancer cell? So we are using spatial analysis, but also an analysis of this interaction so that we can figure out how the fingerprint of a cancer cell is properly received by the immune cell and use this as a biomarker to identify patients where this process is going well versus where it's not going well. The timing and sequencing of therapy is extremely important in diseases like glioblastoma because the patient's tumor, it undergoes massive evolution from when the patient presents himself or herself in the clinic for the first time until they finish their treatment trajectory. So it's very important for every single patient to tailor both the timing and the schedule of immunotherapy. So what gives me hope in our fight against this incredibly complex disease is the recent change in the ideology of the field. Previously, the field was busy importing conclusions and concepts from other cancer types and hoping that if they just throw spaghetti at the wall, that it will solve the problem. This has changed now. They have understood that we have to solve the problem of glioblastoma from the angle of the complexity of the disease itself. And this gives me a lot of hope because this has a right chance of finding a solution. The collaboration between basic science and clinical science is extremely important because clinical science inherently is correlative in nature. Phenomena A and phenomena B are happening together, so they must be connected. This is a little bit way clinical science works, but in basic science, you really have to establish the link between them. You have to prove that phenomena A was either leading to B or not. And this is where the challenge is, but this is also where the meat on the bone is. Because if you can prove by putting these two fields together that A leads to B, rather than that they just coexist, you can really create new biomarkers and therapies. Artificial intelligence systems biology will be very important to do things faster and in a more organized way than a normal human can. There's a huge amount of data involved coming out of these patients, and it's not humanly impossible to do the organization and digestion of the data. It will simply take us too long to reach the same point where systems medicine and AI can help us reach faster. So my view is that it's going to help us organize and digest this data fast So humans can actually do what they are good at, which is interpreting this for the patients. So we have actually found that the traditional view of dendritic cell vaccine, that if you just give it to the patient, that it would create the momentum to help the patient, is not entirely true. Apparently, the vaccines themselves produce pathways that could be making the job of creating this momentum harder. So I'm very excited that we have understood this, because this was underappreciated previously. And now we are obviously applying this logic in the GLIOMATCH project, trying to understand which patients are gaining immediate advantage from the vaccines and which patients are actually creating these counterintuitive pathways. So I'm very excited that we are taking this step of going from basic sciences to clinic so fast, because I think this will really help patients a lot, because that's how complex the disease is. So for example, glioblastoma as a disease is very heterogeneous across patients. So this heterogeneity means that the disease across patients will not behave in the same way, and within the patient will also not behave in the same way. If we do not pre-identify this behavior, then we are not likely to understand where the patient will evolve after we have given a vaccination. So what I'm excited about is that we are already able to now use the information available to us to understand patient trajectories. By getting new information in this project, we will be able to improve this. So this way, the vaccination can be done tailored to each patient and the disease of that patient, rather than being given to all patients without making any differentiation.